Dear students, in today's class, we are discussing about the topic types of egg. First, let us see what is an egg and what are the important functions of egg. The egg is the female gamete which is called ovum. It contains all the materials which are essential for the development of the future embryo. The egg has three important functions. First, the egg supplies haploid set of chromosomes to the future embryo. You know that the egg or the female gamete and the male gamete sperm will fuse together to form the zygote. The zygote is diploid and it is having two haploid sets of chromosomes. Out of these two haploid sets of chromosomes, one haploid set of chromosome is contributed by the egg. So that is the first function of the egg. That is, the egg supplies a haploid set of chromosomes to the future embryo. Then the second function of egg is that it contributes the entire cytoplasm to the embryo. The cytoplasm of the zygote is formed of the cytoplasm of the egg. Then the third function of the egg is that the cytoplasm of the egg stores and supplies food to the developing embryo. You know that for the development of the embryo, it requires nutrients, and these nutrients are stored in the egg cytoplasm. These are the three important functions of egg. Now, what is yolk or deutoplasm? I told that in the egg cytoplasm, the nutrients are stored which are required for the development of the embryo. And these nutrients which are stored in the egg cytoplasm is called the yolk or deutoplasm. The yolk or deutoplasm is formed of proteins, phospholipids and neutral fats. The yolk is classified into two types based on the amount of protein or fat present in it. If the yolk contains more amount of protein, then it is called protein yolk, and if the yolk contains more amount of fat, then it is called fatty yolk. Now coming to the classification of eggs. The eggs are classified into different types based on four different criteria. That is, amount of yolk, distribution of yolk, presence or absence of shell, and type of development. First, let us see how the eggs are classified based on the amount of yolk. The amount of yolk that is stored in the egg cytoplasm depends on whether the development of the embryo takes place externally or internally. In the case of external development, the egg has to store a large amount of nutrients in the form of yolk in the egg cytoplasm. Whereas in the case of internal development, the egg cytoplasm need not contain any yolk. Why? Because the nutrients for the development of the embryo can be obtained from the blood circulation of the mother, because the development of the embryo is taking place inside the womb of the mother. That is the advantage of internal development. But it is not the case in external development. In the case of external development, the development of the embryo takes place outside the body of the mother, and the nutrients for the development of the embryo has to be stored in the egg cytoplasm. The amount of yolk is also dependent on the duration of development. In the case of some organisms, there is a larval stage in the life history. In such cases, the egg cytoplasm need to contain only small amount of yolk. Why? Because there is a larval stage in the life history, and this larva can find food for its development. So the egg has to store only a small amount of yolk which is needed to support the development up to the larval stage. But in most other organisms, there is no larval stage, and the entire process of development has to be completed inside the egg itself. In that case, a large amount of nutrients have to be stored inside the egg cytoplasm, which can support the entire process of development until hatching. Based on the amount of yolk, the eggs are classified into four different types: alecithal egg, microlecithal or oligolecithal egg, mesolecithal egg, and macrolecithal or megalecithal egg. In the case of alecithal egg, the development is internal and the embryo receives nutrients from the body of the mother. Hence, in such animals, the egg contains no yolk and it is called alecithal egg. For example, the egg of eutherian mammals or the placental mammals are alecithal because the development is taking place inside the body of the mother and nutrients are obtained from the body of the mother. So there is no need to store yolk in the egg cytoplasm. Then the second type of egg based on the amount of yolk is microlecithal or oligolecithal egg. When the egg contains small or negligible amount of yolk, it is said to be microlecithal or oligolecithal egg. For example, the eggs of marine animals like amphioxus, tunicates, echinoderms, all these eggs contain a little yolk because the young one that hatches out begins active feeding at an early stage. So there is a larval stage in the life history. The egg has to store only a small amount of yolk. That is microlecithal or oligolecithal egg. Then the third type of egg is mesolecithal egg. In the egg of amphibians and some fishes, the amount of yolk present is only moderate. Such types of eggs are called mesolecithal eggs. Then the last type of egg based on the amount of yolk is macrolecithal or megalecithal egg. When the egg contains large amount of yolk, it is said to be macrolecithal or megalecithal egg. In such cases, most of the portion of the egg cytoplasm will be accumulated with yolk and there will be only a small amount of yolk-free cytoplasm which contains the nucleus. Here there is no larval stage and the entire process of development has to be completed within the egg itself. That is why the egg cytoplasm is loaded with large amount of yolk. Examples of such eggs include the eggs of reptiles, birds and egg-laying mammals. Now coming to the second criteria of classification, that is distribution of yolk. First is isolecithal or homolecithal egg. Here, the small amount of yolk that is present is uniformly distributed in the egg cytoplasm. For example, in the egg of amphioxus and echinoderms, the egg cytoplasm contains a small amount of yolk and it is uniformly distributed in the egg cytoplasm. The next type of egg is telolecithal egg. Here, the yolk is accumulated in the vegetal hemisphere. The egg has two hemispheres. The upper hemisphere is called the animal hemisphere and the lower hemisphere is called vegetal hemisphere. The telolecithal egg is of two types: moderately telolecithal egg and extremely telolecithal egg. In the case of moderately telolecithal egg, the vegetal hemisphere will be occupied by yolk-laden cytoplasm and the animal hemisphere may not contain yolk. Example, the egg of amphibians. In the case of reptiles, birds and egg-laying mammals, the egg cytoplasm is heavily yolked. Here, the vegetal hemisphere is completely packed with yolk and most of the animal hemisphere cytoplasm is also packed with yolk. And there is a small amount of yolk-free cytoplasm which is located near the animal pole and it contains the nucleus. So, in the case of reptiles, birds and egg-laying mammals, the egg is extremely telolecithal. Now coming to the third type of egg based on the distribution of yolk, that is centrolithial egg. In the case of centrolithial egg, the yolk is accumulated in the central cytoplasm and there is a peripheral thin layer of yolk-free cytoplasm which contains the nucleus. The eggs of insects are examples for centrolithial egg. Now coming to the third criterion for the classification of eggs, that is presence or absence of shell. Based on the presence or absence of shell, the eggs are classified into two types, cladoic egg and non-cladoic egg. The eggs laid on dry land must be protected well. Hence, they are covered by a calcareous shell. Such eggs with a calcareous shell are called cladoic eggs. Example, eggs of reptiles, birds and egg-laying mammals. The non-cladoic eggs are not protected by calcareous shells. This type of egg is laid by animals whose development is internal and also by animals who lay eggs in water. Example, egg of placental mammals, amphibians and fishes. Now coming to the last criterion for the classification of egg, that is type of development. According to their potentiality of development, the eggs are classified into two types, mosaic or determinate egg and regulative or indeterminate egg. In the case of mosaic or determinate egg, the fate of each portion of the egg cytoplasm is predetermined. So, if a particular portion of the egg cytoplasm is removed or if it is damaged during development, then a defective embryo will be formed which is lacking a particular organ. Here in this picture you can see that during development one of the cells is pricked with a red hot needle and that cell was damaged and a defective embryo was formed. Such eggs where the fate of the egg cytoplasm is predetermined, they are called mosaic or determinate eggs. Example, egg of annelids, mollusks and ascidians. In the case of regulative or indeterminate egg, the fate of each portion of the cytoplasm is not predetermined. Hence, during the process of development, if any damage occurs to the egg cytoplasm or if any cell is destroyed, then the remaining cells can reorganize or they can regulate development in such a way that a complete normal embryo without any defect is formed. In this picture, you can see that as development is proceeding, the four cells are separated and they were allowed to continue development. And these four cells, they gave rise to four complete normal embryos without any defect. How it was possible? Because even after separation, the cell was able to maintain its potentiality to undergo development. So the fate was not predetermined, it was able to regulate development so that a complete normal embryo was formed. Such types of eggs are called regulative or indeterminate eggs. Example, egg of vertebrates. With that, we finish the topic types of eggs. Thank you.